Welcome to our first week of lectures. So, as all of you know, kind of a refresher from last week, but we're planning to hold weekly meetings throughout the school year to prepare for the USABO. These lectures run into February, I believe, and that's when we will start having our mock exams, so that you guys can have a feel for the actual exam. But for now, they're just weekly lectures. Again, they're in the format of slides, so you guys probably can see the slide now that's on the screen, being screen shared by Veda. This is going to be our first lecture. For lectures, usually we would have a reading from the Campbell's biology book, a few chapters that you guys could read before, but since today we didn't send it last week, then it's fine. But today's lesson, I think, covers parts of chapters 2 to 6. It's not entirely there, so if you guys want to either refresh your memory after reading it or just find more details from the book, then you can read through the Campbell's book. And yeah, if you guys have any questions throughout the meeting, you can throw them in chat. Yeah, so I guess I'll hand it off to Veda now.
Hi guys. Today we'll be talking about cell biology and the structure and function of cells. And this lecture is basically a two-part lecture, so we're going to be continuing this topic next week as well. And today our main focus is on the macromolecules, enzymes, and cell organelles. So let's jump right into it. Okay, so before we talk about life's four biological molecules, it's important to note that macromolecules are basically polymers built from monomers. So what that means is that these macromolecules or big molecules are made from repetitions of smaller pieces, which are called monomers. And the prefix mono means single, and then poly means multiple. So carbohydrates, proteins, and nucleic acids are polymers, which are chains of monomers. And the process by which the monomers become polymers are called dehydration reactions or dehydration synthesis. And this is basically when a water molecule is released to join two molecules together. So in the picture on the right, we can see that there's a dehydration synthesis reaction shown in the top, and there's a water molecule being lost, and then there's two molecules that join together. The opposite of dehydration reactions is hydrolysis, which is basically the reverse, and it is the breakdown of polymers, and a water molecule is basically added. And so these polymers will have a water molecule added to them, and they'll break down into monomers. And a really important point over here that I want to make sure that everyone understands is that there's a really small set of monomers which make a diverse set of polymers. So it's just that we start off with like a few number of things which can, through combinations and permutations, make a diverse set of things. So an example is like DNA has only four nitrogenous bases, and we can see how diverse those bases interact with each other to form the diversity we have. Okay. Okay, so out of the four biological molecules, macromolecules, the first one that we're going to talk about are carbohydrates, which are basically sugars. And carbohydrates are the fuel or the building material, and this is basically what your body uses when it needs to perform metabolic reactions. Um, So the defining features of sugars are that they have one carbonyl group and multiple hydroxyl groups. So as we can see in the picture to the right, we can see these OHs, which are basically the hydroxyl group, and those define the sugar. There are three main types of sugars. There are monosaccharides, disaccharides, and polysaccharides. Um, Monosaccharids are the simple sugars using the same prefix as before, mono, and also called single sugars. And the picture on the top right um, it shows a few examples of monosaccharides. So there's glucose, fructose, and galactose. These are the simple sugars that combine to form, the more complex ones. Okay, moving on. There are disaccharides, which are basically when two monosaccharides are joined together with a covalent bond. And to refresh your memory of a covalent bond, a covalent bond is when the electrons of two atoms are shared. So examples of disaccharides are lactose, which is the combo of glucose and galactose, and sucrose, which is table sugar, and the combo of glucose and fructose. So that's the picture that we see on the bottom right. And again, these sugars are going through dehydration synthesis to form disaccharides and polysaccharides. Okay, so then we have polysaccharides, which are polymers joined with glycosidic linkages. And this is basically a bunch of sugars, not just limited to two like disaccharides. And the thing to remember about polysaccharides is that they are basically storage material, and that's where we get like all our energy from. So the four main types of polysaccharides are cellulose, starch, glycogen, and chitin. And in the next slide, I'll be talking more about these polysaccharides. So cellulose and starch are two polysaccharides that are found in plants, whereas glycogen is found in animals or humans. And glycogen is the polysaccharide that the liver starts breaking down when blood sugars blood sugar levels decrease. So the difference between cellulose and starch is that a cellulose has beta glucose linkages, while as starch has alpha glucose linkages. And starch is again divided into two groups, uh, amylose and amylopectin. And the difference between those is that there are no branches in amylose, whereas there are branches in amylopectin. Okay. Okay, so the next type of macromolecule we'll talk about are lipids, which are basically fats, and they're hydrophobic, which means that they fear water and are not water soluble. Lipids are the only macromolecules that are not polymers built from monomers, because the defining feature of polymers built from monomers is that these monomers are the same, and in lipids it's not like that, because there are different parts to a lipid that are not exactly the same. Yeah, so they have different components. There's hydrocarbons, which store a lot of energy. And the first type of lipids are triglycerols, or fats and oils. And as the name suggests, these triglycerols are basically one glycerol molecule and then three fatty acids, which are attached to the side. And we can see on the picture to the right that this blue rectangle on the left is the glycerol molecule, and then we have three triglyceride molecules to the right. So diving deeper into the triglycerols, in order for something to be a fat, it has to have the glycerol and the fatty acids. And fats are divided into two groups mainly: the saturated fatty acids, and the unsaturated fatty acids. The unsaturated fatty acids are the ones that they have double bonds. And so these are the healthy ones, and they come from plants and fish. And then the saturated fatty acids do not have double bonds, and they're from animals and poultry. So again, there's also trans fats, which are like straight off really bad fats. And again, the point to remember here is that fats store energy. Okay, so the second type of lipids, sorry, are phospholipids, in which there's a phosphate group and two fatty acids present. And so phospholipids are the ones that make up the lipid bilayers of cell membranes. So these are basically, as we see on the picture to the right, there's that pink circle is a phosphate group. Inside the pink circle, the tan circle is a phosphate group, sorry, and then there's a glycerol, and we have a saturated fatty acid and then an unsaturated fatty acid. So basically, in a phospholipid, there's a hydrophilic part and a hydrophobic part, and this molecule is what is used in soaps, because basically the hydrophilic part is water soluble and the hydrophobic part is not, so they can clean out everything that's, like clean out the water and the dirt. So the diversity of the fatty acids, the phospholipids, is due to the different types of groups attached to the phosphate head. So the main part of the phospholipid is the phosphate group, and then the two fatty acids are the ones that differ from phospholipid to phospholipid and create the diversity of phospholipids. And then we have our third group of lipids, which are steroids. And steroids are basically defined with having four fused rings and attached chemical groups. Examples of steroids include cholesterol. We also have steroid hormones. And again, these are characterized by their attached chemical groups. So as we can see over here, in cholesterol and cortisol, there are four fused rings, and then we have the attached chemical groups. Okay, the third type of macromolecule is proteins, and the monomer of proteins are called amino acids, which I assume that you guys are all familiar with. Amino acids are basically, they have an amino group, a carboxyl group, and an R group, which is the side chain, and this is a part that characterizes the amino acid. The amino acid is basically the result of translation in the ribosome, and these amino acids attach to each other and make the protein. And the different types of like the order in which these amino acids attach and the formation in which they attach is what creates different types of proteins. So there's 8 main types of proteins. There's enzymes, storage proteins, hormonal proteins, contractile and motor proteins, defensive proteins, transport proteins, receptor proteins, and structural proteins. So proteins are basically the molecules that help make things happen, because they're really involved in the function of the body, and without them, nothing can really happen. Okay. So there are 20 types of amino acids, and it's really important to memorize all of these and their chemical structures and whether or not they have non-polar side chains or polar side chains, and if they have electrically charged side chains, because I know that in past USABO questions, they focused on these amino acid related questions. So yeah. In the non-polar side chains, the way to remember the polar side chains is that they have hydroxyl groups attached. So you see the OH, HO, OH, or the NH2 in the asparagine and glutamine. So again, as I mentioned earlier, this R group or side chain is what is defining the amino acid. So that's why that's the part, or the R group or side chain, that is highlighted in all of these amino acids. So these are the polar ones, and then glycine, alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, and proline are all the non-polar ones. And then we have the acidic ones. aspartate, and glutamate. And then we have the basic ones, which are lysine, arginine, and histidine. Okay, the last type of macromolecules are nucleic acids, which, as many of all know, are DNA and RNA, the main ones. Um, Nucleic acids, they are the ones that transfer the hereditary information from parent to offspring, and they have a phosphate backbone, usually a sugar and a nitrogenous base attached. So once these three components are there, we can confirm that it's a nucleic acid. And the sugar is the one that differs between the DNA and the RNA. Um, in deoxyribonucleic acid, which is DNA, the sugar is deoxyribose, while in ribonucleic acid, which is RNA, the sugar is just ribose. The nitrogenous bases, three of them are the same in both DNA and RNA, which are cytosine, guanine, and alanine. No, not alanine. Sorry, adenine. In DNA, it's thymine, while in RNA, it's uracil, and all five bases are shown here to the right. Just to refresh your memory on the transcription translation process that makes the protein. Again, so there's the DNA, which transfers info to the pre-mRNA, the mRNA, the RNA, and then the protein, which happens in the ribosome. Again, these nitrogenous bases are divided into purines and pyrimidines. The purines are the adenine and guanine, and the pyrimidines are the cytosine, thymine, and uracil. The adenine and thymine bond, and the cytosine and guanine bond. The way I remember this is kind of trivial, but A and T, there's no curves in their letters, so that's why they bond, and then C and G, they're kind of curvy letters, so that's why they bond. And then just a side note, the bond between cytosine and guanine is shorter, and it's a stronger bond, meaning it takes a higher heat capacity to break. Additionally, these nitrogenous bases bond in hydrogen bonds. Yeah. Okay, and then this is just a picture of ATP and the three components that make up ATP, which is adenosine, ribose, and triphosphate, the three negatively charged phosphate groups. Okay, so before we go to our Kahoot, I wanted to give just one of the usable questions from, I think this was a 2006 test. So whoever knows the answer can raise their hand, and I'll give you guys some time. Joshua, is anyone raising their hand?
Doesn't look like.
Okay. Okay. I'll give you guys until 4:56. Oh, I see a raised hand. Uh, Sai, do you have an answer?
I'm not really sure, but is it antibodies?
ATP?
No, antibodies.
Oh, no, that's not the answer, but good attempt. Does anyone else want to try? Okay, cool. I'll just show you guys the answer. So the answer is steroid hormones. And I'll explain this answer really quickly. So the question is, which of the following molecules is able to cross the lipid bilayer? So as I mentioned earlier, the lipid bilayer of the cell membranes are made of phospholipids, the second type of lipid we talked about, and um, basically all the molecules that can pass freely through the lipid bilayer are molecules that are lipophilic, meaning lipid soluble, or hydrophobic, which is the opposite, which is afraid of water. So out of all of these, um, antibodies, insulin, and sodium, I think, are all hydrophilic and not hydrophobic. So they're all water soluble, which is what makes them unable to pass through um, the lipid bilayer without support from receptor proteins. ATP, I think, has hydrophobic and hydrophilic um, sides to it, so it's not the best option. But steroid hormones are examples of lipids, which is what makes them lipophilic or hydrophobic, and they can pass through the lipid bilayer quite freely. So yeah, that concludes my lecture. Um, Joshua, if we can move on to the Kahoot.
Yep. You can share it, right?
Yeah,
Right.
okay. Okay, you guys can go ahead and join. The game pin is 2117916. Okay, I'll go ahead and start it. Is anyone still waiting to join? Or is everyone in?
We have around 15 or 14 people now in the um, meeting, so maybe some people still haven't joined.
Okay, are the officers gonna join as well?
Some of us are, I think.
Okay.
No.
Okay, I'm starting it. Good job, guys. Okay, so most of you got it right, but let me just quickly explain why the other answer choices are wrong. Um, a type of amino acid, that's recall that amino acids make up proteins. Steroids are actually a type of lipid, so that's a completely different biological macromolecule. And then again, a type of sugar is a carbohydrate, so that's another macromolecule. And then things that get athletes in trouble is just not the scientific answer I was looking for. Okay, so most of you got it right. Hydrocarbons are a source of energy. We can quickly go back to the slideshow and I'll show you guys. So here. Yeah, so over here we can see that hydrocarbons are entirely made up of hydrogen and carbon, and they're in the lipid group, and um, they basically are a source of energy. Okay. Sorry guys, I clicked the next button, but um I'm not sure if you guys got that correctly. We can just move on. Does anyone have questions about that? Okay. Okay, so most of you got it right. Again, the dehydration synthesis part, the dehydration synthesis is the right answer. Hydrolysis is the opposite of dehydration synthesis. And the way to remember which one does what is when there's the creation of a larger molecule, just remember that synthesis means creation. So that's why it's called dehydration synthesis. And dehydration because you're losing that water molecule to create um, a polymer. And hydrolysis is the opposite. So that's when you gain a molecule and you break up. Okay, wow, good job, guys. Yeah, so cellulose is from plants and it has beta-glucose linkages. The other answers are all incorrect because glycogen is not from plants, it comes from animals. Starch is not from plants and it's from animals. It's not from animals and it's from plants. And then amylose, which is one type of starch, comes from plants, but I don't think it's B glucose linkages. Okay, good. So monosaccharides are not complex sugars. Remember the prefix mono means single or simple. So yeah, the answer should be that they are simple sugars. So that's why this is false. Disaccharides or polysaccharides are um, the combos of multiple monosaccharides. So that's where the complexity of the sugars comes in. Okay, so for this one, I think I could have worded the question a bit more clearly, but I was looking for the phospholipids, and those phospholipids have a phosphate head, and phospholipids are the ones that make up the um, cell membrane, so the lipid bilayer. So yeah, carbon head, nitrogen head, and lipid head. These are just not not phosphate heads. So okay, so this was a bit of a tough question, but I think it's important to memorize the this table right here. Um, so the methionine falls under the nonpolar side chain because again it doesn't have the hydroxyl group, the OH group. So that's why it is true that methionine has a nonpolar side chain. Okay, last question. Okay, so glycine does not have a polar side chain. Again, looking back to this table, um, glycine falls under the nonpolar. It's the first nonpolar side chain amino acid. So yeah, again, it doesn't have the hydroxyl group or the uh, H2N group that asparagine and glutamine have. Okay. Yeah, that concludes my lecture. Do you guys have any questions that you want to ask? Okay, I'm taking that as, oh, okay, maybe. Oh, right. Okay, sorry. I forgot to show winners. Um, 1 second, guys. Okay, great job, guys.